Okay, welcome to session 6, our second to last topical lecture before I have all of you do presentations on during session 8. So we're going to cover today clustering, which is just the first section of the basic machine learning chapter in the textbook. As well as a bit on the section of batch effects, chapter 9. Just a quick review of our last class on distances. Remember that metrics, for something to be a metric, there were strict requirements. There were 6 of them, and I have seen in several papers, actually even looking back at one that I was a co-author on, things being called metrics that were not actually metrics, which is okay generally for our purposes. Something can be a distance with only 3 of those 6 requirements and a similarity or dissimilarity measure that have fewer yet. And this is what generally we will care about for the purposes of clustering. And the only real requirements for similarity and dissimilarity metrics are the non-negativity requirement and the symmetry. So either the similarity or dissimilarity, we're generally going to use dissimilarity, which is similar to a distance, in that it is always positive. The dissimilarity between me and you is the same as the dissimilarity between you and me, and dissimilarity will increase as 2 objects get more dissimilar to each other by some measure. And there are different ways that we can define similarity and dissimilarity, and this is a somewhat it's putting a quantitative formula on something that is somewhat qualitative, in that the way we will try to define a dissimilarity measure that somehow corresponds with how we think of our measurements being similar or dissimilar. And we'll the 2 main ones that we often talk about in these classes are Euclidean distance and 1 minus Pearson correlation. Those are very common ones for continuous data, but there are other ones commonly used for qualitative data and for discrete categorical data, such as DNA sequence data. Let's recall this tissue expression data set that comes from the website of this class. It had 22,000 rows, 100 corresponding to probe sets for genes on a microarray, and 189 columns for samples. And just as a note about, this class uses, the textbook uses microarray examples. In your projects, you have an RNA-seq example. The main is that the RNA-seq data needs to be transformed in order to produce results similar to what you would get from a microarray. The issue is just that with the count data, it has a very, very long tail, and measurements like Euclidean can be dominated just by a single gene, because you have so much more dynamic range than you do with a microarray, that having a large amount of expression of one gene can really dominate the distances, which usually is not what you actually want for clustering and dissimilarity. So I've done quite a bit of comparisons between in TCGA. There are often you have both. Come on in. Sorry. It's okay.
Good morning.
Good morning. In the TCGA data sets, we often have both microarray and RNA-seq data, with the RNA-seq data summarized at the gene level, and at the level of whole transcriptome distances, they're very consistent with each other after taking log transform of the RNA-seq data. Okay, so we have 189 samples, and we also have this tissue variable that says which tissue they came from. We have 7 different tissues, and between 6 and 39 of each of them. So in the last class, we calculated distances on these samples, and using this dist function to calculate Euclidean distances. And just to give you a bit of preview here on motivation for clustering, here is a heat map, which is with where I'm plotting actually these Euclidean distances. So rather than a typical heat map that might show gene expression itself, it's it's plotting the distances themselves. So these do not represent expression of genes; they represent pairwise distances between samples. And I've used hierarchical clustering here to gather together like samples. You see along the diagonal, these are values of 0 because the distance between a sample and itself has to be 0. And you see groups of pretty similar samples with very, very small distances relative to other tissues. So we've managed very quickly here to create a map that shows like tissues, or we don't know for sure yet whether they're like tissues, but it's certainly placing together samples that have similar expression profiles. And using the other type of measure that we introduced, 1 minus the Pearson correlation, where I use as.dist instead of dist here because I just wanted to directly convert this matrix into a distance object without calculating, doing a further distance calculation on this matrix. And it looks somewhat different. The scales are different, but it is still putting together, well, we have kind of 3 medium-sized groups and then some smaller ones, maybe a 4th. It does look a little bit different on the correlation, using the correlation distance measure. So this gets at how the results of clustering can depend on the distance metric or the distance measure that you use. One advantage to using this 1 minus Pearson correlation is that this is really directly interpretable, this scale. So a value of 0 means that we have a correlation of close to 1, almost perfect correlation among some of these samples. This is, I think, around 0.99 correlation between different samples of the same tissue. But at the very lowest correlation, we're down to a distance of 0.4, meaning a Pearson correlation of 0.6. So even though they're from different tissues, they're still human cells. They're made up of, they all have some stromal cells. They all have high expression of some housekeeping genes. There's also here some control probes for the housekeeping genes, which will be highly expressed in every tissue. So, so even totally different tissues have some relationship to each other. So how do we do that clustering? That's what we call hierarchical clustering. Now, all clustering methods use a dissimilarity measure between observations to try to group together similar observations. and to form groups of similar data points, whether those data points are samples, tissues, patients, or whether they are genes or variables. It's going to use your dissimilarity measure to try to group together like things. And hierarchical clustering is just one of many, but it's a very commonly used one. I think, in my experience, the most commonly used. Two main approaches, or two possible approaches to hierarchical clustering. The first one is what is almost always used, probably because it has the most convenient tools for it, and it also produces clusterings that are familiar to look at. And by bottom up or agglomerative, what it means is that you start out with every point in its own group, and then you start by grouping together the most similar 2 points, and there's your first group. And then you group together the next